Hello everyone, and welcome to our TERN webinar for the 7th of September on technology for conservation traces of biodiversity. A few technical problems. So we at TERN acknowledge the traditional owners and custodians throughout Australia, New Zealand and all nations, and we honour their profound connections to land, water, biodiversity and culture, and pay our respects to the elders, past, present and emerging. And today I'm talking to you from the Tjabukai country in far north Queensland, amongst the rainforest people. Please feel free to put into the chat where you're from if you have any questions and answers, questions that you want us to answer, put them into the Q&A for the event. So TERN is the Terrestrial Ecosystem Research Network, one of the major research infrastructures of Australia monitoring changes to the environment. And we wouldn't be possible if we weren't partnering with so many different groups across Australia. And it's really good to be able to mention those today. Today's talk, we've got 4 speakers. We've never had 4 speakers in an hour before, but we're going to have presentations from Dr. Emma Spencer, who's from World Wildlife Fund Australia, from Dr. Miles Mentz, who's from James Cook University, from Alison Malpatida, who's from Charles Darwin University, and Associate Professor Susan Fuller, who's from the Queensland University of Technology, despite what it says on that slide. So I'm not going to go through their biographies because you have them to read. But before we just commence, I want to say thank you everyone for joining us today. We've got an outstanding number of people who have registered. I'd like to also mention that the chair of the TERN Advisory Board, Hugh Possingham, has joined us today. Welcome, Hugh. And welcome to anyone else that is from the TERN community and board and everyone else. So I'm going to hand over to Emma, who's going to tell you a little bit about camera trapping. So over to you, Emma.
Thanks very much, Beryl. I'm going to just set up my screen. It's quite lucky. My computer decided to do a full reset without telling me about it just then. So luckily I signed back on just in time. Right, all good. Okay, so today I'm going to be talking about Eyes on Recovery, which is a WWF-led and Google-supported large-scale collaborative monitoring program that's using camera trap technology as well as machine learning and artificial intelligence to essentially assess the impacts of the 2019 and 20 bushfire season. Now, before jumping into this presentation, I just wanted to begin by acknowledging the traditional custodians of the land throughout Australia. Now, because this program is spread across so many regions, I didn't mention every group here, but I do want to specifically acknowledge the Gundungurra, Darug and Wiradjuri peoples of the Blue Mountains area, whose land it is that I'm currently calling in from. Right, let's get stuck into it then. So the Eyes on Recovery program basically arose in the wake of the 2019-20 bushfire season, where millions of hectares of bushland was torched and where billions of animals were displaced or killed. Now, in the wake of the fires, it was somewhat unclear about how our native species were recovering, especially some of our threatened or iconic species like this cute little koala here. Now, in addition to this, understanding wildlife recovery and impacts at this time, it seemed to be this almost insurmountable task, largely due to just the massive scale of this event. So that's really where Eyes on Recovery as a program came in. So this program was built to be as collaborative as possible. As one organisation couldn't hope to get out there and monitor all the areas that were impacted by these fires. So at WWF, we partnered with Conservation International and we found funding and technology support from Google to develop the Eyes on Recovery program. We then reached out to a huge range of organisations, from universities to community groups to government organisations, to essentially deploy hundreds and hundreds of camera traps out in fire-affected regions in Australia, and to capture wildlife data, what was happening, you know, in terms of their recovery. Now, a key issue with camera traps is the sheer amount of images they can generate. And we're talking, you know, millions and millions of images that need to be sorted through. You've got to sort out images without animals. You've got to go through and identify the species within images that do have animals. So our solution to that big problem was essentially to partner up with Wildlife Insights. Now, Wildlife Insights, I don't want to go into too much detail here. You can, it's accessible online. You can look it up, just search up Wildlife Insights on Google. But essentially, Wildlife Insights is an online platform which allows users to upload, you know, the millions of images they might have, and store those images online, store that metadata for those images, you know, where the image was taken, the methods that were used to set up the cameras. It also helps with the identification side of things, or the image processing sort of side of things. So it implements AI and machine learning to basically assist users to really rapidly filter out any blanks, so images without animals present, and also to identify those species within the images that do have animals present. Finally, and although this is under development still, but it also provides sort of an analytics side. So it also allows users to, you know, get out there and understand what their camera trap data actually means, providing measures, you know, such as species diversity, species richness, activity, occupancy at a site, those sort of things. So it's a really good platform. And I'll also say that all the data that goes onto it eventually ends up as, you know, it's being open source, so you can access it and understand, you know, what's happening in Australia across a whole bunch of different places, or even globally, because it is a global platform. I'm going to go back now, back to the program, Eyes on Recovery, and what we've actually been able to do so far with this program. So, and I wanted to kind of use this to, I guess, illustrate the extent and the scale of this program as well, because like the fires, it had to be a really massive project in scale. So far we've joined up to over 20 partners. So this has included, you know, university groups, NGOs, and 4 state government agencies. We've almost got about 1,200 cameras deployed across about 9 regions at this point. I've just illustrated those regions on the map there for you. And within each of these regions, there's, you know, it's not just one project per region. Some of these regions have several projects in them. So we have 17 different projects running across these regions. And just so you understand, those little circles are the regions, and the numbers in them are the number of camera traps that we have out in those sites. So far we've generated more than 4 million images, and they've all been uploaded to the Wildlife Insights platform. And, you know, this is just a start though, because we're bringing in more and more images every day. So there's a lot going on. Obviously, I can't go through all of the projects. We have 17 of them, but I just wanted to provide, you know, I guess a little bit of a taster there in this map. Some of these projects are looking more at fire management practices like cultural burning, and we've got an Indigenous-led project up in northern Queensland with NQ Dry Tropics, looking at, you know, the impacts of cultural burning practices and such. From that management side of things in the Blue Mountains, we're also looking at things like fire severity and fire frequency with the University of New South Wales, and a student-led project, which is sort of going to help to build on our understanding of how different fire regimes impact the species we see in different locations. And then we also have a range of projects that are very species specific, sort of generally focused on endangered or threatened species. So we've got a great brush-tail rock wallaby project with National Parks and Wildlife up north in the north coast sort of region of New South Wales. And we also have a great project on Kangaroo Island with the Kangaroo Island Done-Up project. And a lot of these projects, it's not just all one, I guess, standardized camera trapping array that we've set up at every single site. because obviously some of these sites we're looking at species that require certain camera methods to be set up. So for instance, a dunnart is a very small animal. You're going to be wanting to put a camera quite close to the ground to pick up an animal like that, compared to, let's say, if you're monitoring predators, you might want to put cameras on the road, for instance. Now, again, I'm not going to have time to go through all the results, but I do want to just highlight that there are some really great stories of recovery coming out from this work. So, for instance, the Kangaroo Island dunnart, this species is only found on Kangaroo Island. 95% of its habitat was burnt in the fires, so massive scale there. And it was thought, are we even going to find any when we put cameras out there? But the really positive story coming out of that area is that almost all of our monitoring sites have picked dunnarts up at this stage. So it's really, really exciting. So I also wanted to highlight that apart from setting all these projects up and assessing fire impacts, this program has also been working to improve the wildlife insight species identification models. So I did mention this before, but Wildlife Insights actually started in the US and it moved global. So most of its species training data, which sort of essentially you upload a whole bunch of images that have been pre-categorized or pre-classed into species, and then you can use that to train your models so that then you can run that model on images that haven't been identified already or tagged to species. And that model can then pick up those different species based on that training data. But essentially, we didn't have any of that training data for Australia. So that's what this program has also been feeding into and contributing to. So far, we've added almost 2.5 pre-classified images through this program to Wildlife Insights. And that's been now feeding into the latest model. So that's including, more than 100 species that are present in Australia. And it's really helping to speed up our project partners and how quickly they're able to process images on the platform and just in general. So that's a really exciting thing that's come out of this project as well. I've just got a bit of an example here on the variability that you can get in terms of the species precision of the model that we've built. So there are some species that are much more common, like your lyrebirds, your brush-tailed possums, where we've got a lot of images of them. They obviously have quite high precision in the models. We can identify those species very easily using Wildlife Insights and the AI machine learning models. But there are other species that are a bit rarer. In this case, maybe the long-nosed potteroo. We don't have quite as many images, and the koala, a bit harder to identify using the models at this point. Now, aside from everything else I've already spoken about, we're also trying to develop some analytics tools. And, I'm not going to go into too much detail on this either, but I did just want to flag that we're also trying to develop this tool, the Eyes on Recovery tool, which we're hoping to release next year. And this tool is essentially going to draw data from our Eyes on Recovery projects and allow these project partners to visualize and understand species data in different areas impacted by different severities of fire, or compared to, say, unburnt areas. As I said, still in development, still taking on feedback from project partners, and we're hoping that it will have other applications, not just in Australia, but globally and for future fire events. So I just wanted to finish up now by, I guess, outlining a few key reflections and gaps that we've gathered so far from this project. And a big thing most fire projects are finding right now is there's just a lack of baseline data. So data before the fires hit. So it's quite hard to actually tell, how are things recovering? What are we missing out on? What have we lost? Because we just don't have that data. We've also identified a real need for more standardized camera trapping monitoring protocols. So that's something that I think we really need to work on as a country. Yes, they might have to be specific for specific species, but it's something we should still work towards to develop. As well, I wanted to flag, machine learning, AI, all of that sort of stuff. It's not yet perfect. We need more training data, but it is doing really well, and it's doing, better than a lot of the offline, the other options right now, which is sorting through everything manually. and all of our online image processing platforms, so they have room for improvement. And finally, I just wanted to flag, there is a real need as well that we include the Aboriginal and Torres Strait Islander voice in all of this, and that's something we're going to be trying to do more of into the future. I'm just finishing on this slide. I know that my time is now up. I'm going to have to hand over to Miles, but I wanted to say that we are trying to address some of those points that I flagged using the Wild OBS camera trap data pipeline. And if you want to find out more about that, I've got a little link down below. I'm happy to take questions later if you're interested in what it is and how it can help us get information from our camera traps to standardized information from camera traps all the way to national database where we can share and understand conservation issues across Australia. So thank you very much, and I will be handing over to Miles now. So
lovely, thanks very much, Emma, and thanks as well to Beryl and for the rest of TERN for asking me to come and give a talk in this. And I'm going to be presenting a little bit about what's known and what we can actually do with radar for monitoring animal movements and monitoring animal populations. So radar is really a key technology that's now being used to remote sense biodiversity. And so it allows us really to observe what's going on in the airspace. And it's been used to sort of really coin a new field of study, effectively aeroecology. And when we talk about radar in this context, I'm talking about everything from marine radars, so typical things that we might see on boats with the classic T-bar antenna, to sort of more specialized entomological radars, which have a parabolic antenna, through to weather radars, which also can be used to observe biodiversity. Radar is really useful for monitoring and forecasting of movements of animals, but it can also really give us an idea of what's happening with big species, so things that might be either flying high up or things flying in the dark. It can be really useful for informing conservation, so it can give us an idea of areas that might be key for animals, for example, en route, or key foraging areas. And also, it can really point out areas where there might be human-wildlife interactions. For example, there's studies that might look at the effect of fireworks on bird movement or disturbance of bird roosts, and also interactions between fishing boats and seabirds. And radar as well, over here on the right, there's this little image. It's a paper we recently did in Tree, and it's been highlighted as one of these new technologies that's actually being used for basically determining or understanding biodiversity. And so this also fits in with computer vision and machine learning. As Emma mentioned, we also can think of acoustic monitoring and also molecular methods like metabarcoding.
Sorry, we seem to have lost Miles for a moment. So if everyone can...
that are going on. We can also look at understanding dispersal of animals, which might end up dispersing pathogens. We can think about pollination, crop damage, pest control, or even conservation areas, and trying to understand animal interactions with their habitats. And so I'll start a little bit talking about weather radar and give some examples about what weather radar has actually been used for in terms of conservation and monitoring. And this figure actually shows the active weather radars across the globe, and overlaid on that are a number of our major flyways. And we can see as well there, we have the East Australasian flyway and the Afro-Palearctic flyway, and there's a huge number of these radars that are actually set up in networks, and particularly in countries like Australia or the US, where we use similar types of radar across the whole country. These can actually be used as a huge remote sensing platform for animals as actually they're on the move. And this has been used really quite successfully in the United States, also in conjunction with citizen science data from eBird, to actually monitor and forecast bird movement. But also we can look at, here's an example of a weather radar study in Europe, where you can actually see the full flux of birds migrating in autumn as they move southwest through the continent and towards Africa. The utility of weather radar is something that's come up mostly in the past few decades, because for many years, biological targets, birds, insects, and bats were basically filtered out because meteorological organizations were more interested in actually the rain and what's going on. And so the biological signals are very much noise, and so they tended to be filtered out. But now they're being extracted and used much more to get this really rich data set on both movement, biomass, and general flux. And while it can also give us an idea of what's happening in terms of movements in general, we can see here these blue spots that are rising in the middle of these figures is actually birds ascending from the Okinawa Islands after stopover. So it can also be used to identify really key, for example, stopover sites, feeding areas, other areas that may be being used by animals. And this can allow us to actually allocate or potentially look at these as being key conservation areas we might need to either mitigate risk. So while birds are fairly large targets for radar, weather radar especially, is also particularly good at detecting insects. And so the figure on the left is these are actually mayflies emerging in the United States. And so these are these mass mayfly emergence where you actually get millions of insects coming on a certain number of nights. And by seeing these huge cluster of mayflies emerge, this has actually been linked with citizen science data to actually look at long-term trends in mayfly populations, which is also really key for understanding waterway health and general monitoring of biodiversity in that context as well. And so the one useful thing that they have in the United States is that they have an X-RAD radar system, which has a database going back for about 25 years. So they can actually look at really long-term patterns. And by linking this with long-term monitoring, they can see that there's been really a huge decline in these waterway insects across the United States. Now I'm going to switch across from weather radar to a slightly more specialized radar system. These are vertical looking radars that are used for monitoring mainly insects. There's some out there that are used for birds. And basically the reportable radar unit, which can be seen in the image in the middle, and these can just be towed around on a trailer and set up with a generator or connected to the mains. And they're often being used, particularly in Europe. There's a network of these now where they're actually positioned in wind farms and they're observing what's going on in the airspace above, and then they can look at where the birds are and what heights they might be at, and is there a collision risk for the wind farms and how can they mitigate that. So they're also being used as a way to even check the airspace before certain developments are actually put up. And there's movement towards using some of these radars as well in terms of managing or mitigating bird strike risk at airports. The bird radars tend to have a broader antenna, so they have a bigger range that they pick up, whereas these entomological radars have a very narrow antenna. So they basically only have a 1 degree beam that comes up, but it means that you get much higher resolution. And so these can actually see quite high into the airspace. This model is one that can detect insects of about 50 milligrams. So we're thinking of a medium-sized moth potentially up to 1 kilometre above the ground, whereas some of the newer models that research groups are actually developing can see about 2 kilometres. And so they have this immense range of detection. And they're also able to detect very small insects, such as aphids, at least within a few hundred metres above the ground. One of the really cool things about these radar systems is that while they spin on their axis, they also have a slight wobble in the antenna, so it's called a nutation in the beam. And what this actually allows is that the signals being detected in the radar, you can actually get an idea of the size and the shape of the object. So not only do you get where it is in the airspace, so the height, you can get size and shape, and you can also get the direction in which it's moving. And so this has actually been used quite successfully, particularly in insect migration, to understand insect movements and migration at certain hotspots, points in the landscape. And for this, I'll give a little bit of an example of what we can do with this sort of thing. And this is from a study that we did a few years ago based on a couple of long-term radar monitoring sites in the southern United Kingdom. And so for this study, we were actually looking at hoverflies. So hoverflies are a really key part of the ecosystem. They're major pollinators, so alongside bees in terms of their amount of pollination that they actually undertake. Also, many of the species have larvae which feed on aphids and other small crop pests. And so they're also able to provide a biocontrol as well. The hoverflies are a really interesting group. So we were looking at long-term movements of hoverflies across the United Kingdom. So first, what we were able to do here is by linking in with citizen science data. So the hoverfly recording scheme is determined that we were looking at these two species as the most abundant migrants, the eukaryotes and an episurface. And we could estimate that there was about 408 million immigrating into the United Kingdom and about 1.5 billion emigrating. So this is the flux of about 32 tonnes of biomass. And so this also gives an idea that the migration is really beneficial for these species in terms of the population, because they come in, build up their numbers, and then move back out. But in terms of thinking about this number of animals moving, is quite interesting, and biomass flux is really important for general ecosystem function. We can also think about this in terms of the services that they might provide or the functions that they have in the ecosystem. And so for this, we were actually able to calculate that that's about 20 hoverflies per square metre of cereals and the transport of about 3 to 8 billion pollen grains. And when we think about 20 hoverflies per square metre of cereals, a single hoverfly larva can eat effectively about 400 aphids before development. Look at all the directions they're moving. So we can see here in the spring, we can really detect that there's a north flux. In the summer, there's not strongly directed movement in autumn. And so we can see that they're just generally milling around and doing what they do. And then in autumn, they turn around and return back south. But on top of that as well, we can also get some idea of how they actually respond to winds. And so this is not just how the flies are moving, but we can actually add in weather data to this. And we can see, for example, in the red dots on the panel A, this is actually the difference between the track that the animals are moving in and their angle of correction, so the heading versus the track. And we can see that they're actually not necessarily correcting their path in response to what's happening with the winds. And that's because in this case, in spring, most of the winds are favourable and moving north. Whereas in autumn, they're actually selecting favourable winds to migrate. And so this is a really useful tool. We can actually get information for forecasting movements, and vertical looking radars are also a key part in the plague locust commission. So they're being used to monitor locust movements and have been doing so in Australia for quite a number of years. And
I think we've lost Miles again as the signal from Europe peters out coming to Australia. I'll just give him a second.
Areas for conservation, but it's also key for we need to establish networks, particularly of these vertical looking radars and not just the weather radars as well, because they give us quite different information. Radars are going to be improved by improved classification algorithms to actually really allow us to see what we're looking at in the airspace, but also they rely quite strongly on ancillary data. So for example, in combination with citizen science, acoustic recording networks, etc. And with that, thank you very much everyone for listening, and I'll hand over to Alison.
Thanks for that, Miles. And thanks to the TERN team for asking me to talk in the webinar today. So my name is Alison Mapatita, and today I'll be talking about my PhD project that aims to assess genomics approaches for monitoring terrestrial invertebrate bioindicators, especially in the context of ecosystem restoration. So I'm currently a candidate at Charlestown University, which is located in the Northern Territory. And I'm doing my research as part of the Research Institute for the Environment and Livelihoods. And my project is funded by the Supervising Scientist branch, which sits within the Department of Climate Change, Energy, the Environment and Water. So why do we do monitoring for ecosystem restoration? We do it to assess whether the restoration has been successful. So it seeks, restoration seeks to assist in the recovery of an ecosystem that has been disturbed, so from agriculture or land grazing, and also from mine sites, back into a healthy ecosystem. So what it looked like pre-disturbance. And you can see in the photos here, I've just got an evaporation pond from an old mine site up in the NT, and what the nearby reference sites look like. So savanna that hasn't been disturbed or impacted by the mining site. There are several limitations to current monitoring, as there is often a vegetation bias for measuring flora over other indicators, such as animals and insects and soil microbial communities, which help the ecosystem to thrive. So it's really expensive to do these surveys, and taxonomic identification requires these specialists that are very hard to come by. And another challenge is that there are a lot of knowledge gaps in ecosystem restoration science, and there are quite often short time scales for project completion. Yet there's still a push for restoration, and there's an urgent challenge to develop early indicators for critical ecological processes that show that the ecosystem is on a trajectory towards restoration. So we use terrestrial invertebrates as bioindicators, and there are several reasons for this. They dominate faunal biomass and play many ecological roles, and they're also highly sensitive to environmental change. Several studies have shown that they're also well-established indicators of restoration. And another advantage is that they're numerous and readily sampled. So we can use pitfall traps, which we put in the ground, and these insects walk over and fall in the pitfall traps. And then we take that pitfall trap back to the lab and do the analysis. So there's some challenges with this type of monitoring, because sample processing can be very time consuming. And it's also very expensive to identify these insects. So the specialist taxonomic expertise is hard to come by. You know, there's generally 1 or 2 specialists around Australia, and they're all located in different states. So it's quite difficult. And also Australian arthropod diversity is quite undescribed, and there are some estimates that this could be as high as 75%. So if we find a species, we know it looks different from others, but it hasn't been formally described, and we don't know how to classify it. So why I'm focusing on ants and termites is because they're both dominant faunal groups. So ants in particular are known as soil engineers. So they move through the soil and build structures such as their nests, which also helps with nutrient cycling, soil turnover, and water infiltration. They're also relatively stationary as opposed to other faunal groups that are transient, like birds and flying insects, which can come from nearby reference sites and come in and get the food and then leave again. So it's not that they'll be recorded as present, but they're not actually using those sites long term. And also ants occur all year round. So termites are often overlooked as bioindicators because they are quite difficult to identify. You generally have to find the soldier termite, which are the termites that have the mandibles, which was in my title slide. And the majority of samples that you'll collect are workers, which don't have any taxonomically identifiable features. Different termite assemblages are also likely to have different ecosystem effects. So you've got wood-eating termites and soil-eating termites, and also grass-eating termites. So depending on what the different assemblage types are, you can get different effects. So on the right here, I've got a ordination diagram of what ants look like at a restored site. So at Ranger Uranium Mine in the Northern Territory. So across time. So as you can see, as the time increased, then you get the species lists becoming closer and closer to the reference sites. So the current method for morphological identification is looking at ants under the microscope and looking at the different differences in morphology, and that's how you can tell what different species are there. So there are new genomics approaches that are emerging, which can help us for invertebrate monitoring. As sequencing technology is getting cheaper and more accessible, we can start to use these technologies more. So they all use next generation sequencing to determine what species are present in the environment. So genetic approaches such as environmental DNA analysis and metabarcoding have the potential to transform how we assess ecosystem health. So what they do is they use species that exist in the environment, and they look at the DNA, and from that DNA, we look at a barcode, which is a species-specific DNA section that will identify what species were present. So it tends to be a more cost-effective approach. There are some estimates that it's about half as expensive as normal morphological identification. However, one of the challenges is that there are limited reference DNA databases, such as the BOLD or the Barcode of Life data systems doesn't have every insect species, and the same with GenBank, which is another reference database. So I just want to go back to metabarcoding. So what is metabarcoding? So it uses these barcodes and it simultaneously sequences tens to thousands of these barcodes in a single environmental sample and gets you back a species list. And I also just want to concentrate on DNA, environmental DNA while I'm on this slide. So eDNA, which you might have heard of before, is a complex mixture of genetic material that can happen from different organisms found in an environmental sample, and you can get it from different substrate types, such as sediment, soil, water, and even air. And this could come from mucus coming off a fish in water, or insects after they've eaten some plant material in the savanna or in the environment, or even from plant pollen that gets distributed through bees. So the reason why this is possible for ants is because my supervisor, Alan Anderson, has been collecting ant barcodes for the past decade, mainly for taxonomically challenging ant genera, and he's got about 10,000 ant barcodes. And for termites, it's a little bit easier because there are less species. So there are about 300 in Australia and about 50 within the NT, and about half of them already have barcoded regions. So that's really good for my project in particular. Whereas all the other insects are, like I said, not very well described, and there just hasn't been that push to get barcodes for all of the different families, like butterflies and grasshoppers, and we're still finding new catchidids, and yeah, NT is very diverse for insects. So I just want to go over to what a DNA metabarcoding workflow looks like. So I have different sample types with my project. So I've got a specimen or bulk sample type, which is on the top left here. So what I do with this is it's a pitfall trap where I get all of the insects and we take it back to the lab and we ID them under the microscope. Once we've ID'd what species exist in that sample, I will then do a DNA extraction where I take out the DNA from those insects, either by blending them up or looking at other methods that can extract the DNA in a less destructive way, and then performing PCR amplification. So PCR amplification basically takes that, say, one DNA molecule and turns it into thousands or millions. So you're getting a snapshot of what's actually in there, but you're amplifying it to such a way that the sequencing technology can then pick up what barcodes exist in that sample. So there are two ways that you could do the sequencing. You can either send it to a commercial laboratory like Ramachodi Centre for Genomics down in New South Wales, or you can get a benchtop sequencer, which is where you have one in your own lab and you hire a technician to do the sequencing for you. So it's a little bit more time sensitive in the second option, because you can get the sample from the environment, take it back to the lab, do the processing, get the sequences. Just to give you an example, if we were to just do morphological identification for these pitfall traps, it could take up to 6 months for one mine site, whereas using the sequencing technology can make that time frame a lot shorter after these protocols have been validated. So after sequencing, you get a species list. So these species lists are created once you match your DNA barcodes back to your reference databases. So you use those two barcodes to make sure that they are the same species. And another way I'm doing it is by using eDNA. So eDNA is can come from soil or from a termite bait for my project. And the soil we're using because ants walk over the soil and they make their nests in the soil. So we're expecting ant DNA to be there. And the termite baits are a little bit trickier because termites tend to burrow quite deep underground and create mounds, but they're not always found near the soil surface. So you use these baits to attract them out of the ground and eat the food source. And then from there, we use a phosphate buffer wash or some sort of protocol to get the DNA off that bait. And then we can get a species composition list of what termites exist in that area. So I just wanted to go through some of my sampling sites. So there's a couple of mine sites up here in the NT. These ones in particular are within the Alligator Rivers region, except for Pine Creek, actually. So Narbarlek is an old mine that's been restored 25 years ago. And it's located about half an hour out of Gumbalanya. It's very remote, very difficult to get to. Lots of water creek crossings, which is fun. And Pine Creek as well, which is a gold mine near the Pine Creek Township, and Ranger. I think I've run out of time. Is that right, Beryl? Yeah. So I'd just like to conclude, biomonitoring of invertebrates is a reliable way of assessing ecosystem restoration success. eDNA and metabarcoding are new genetic approaches to do this and will be more prevalent in the future as this technology gets cheaper and cheaper. It increases accessibility for researchers and industry specialists to evaluate restoration. And my contact details are there. Thank you very much. I'll hand over now to Susan Fuller.
Okay. Thanks very much, Alison. Always lovely to see young researchers being invited to present at things like this. So job well done. I'm here today, Susan Fuller from Queensland University of Technology, the Faculty of Science there. I'm here with a team behind me, obviously, a lot of great people who've contributed to this talk, so I'll acknowledge them when I get to the end. I'm going to talk about acoustics, which is my favourite thing, second only to conservation. So I'll get started. I would like to acknowledge the traditional owners where QUT stands, the Turrbal and Yugara as the First Nation owners. We pay respects to their elders, laws, customs and creation spirits, and we recognise that the lands where we are undertaking our research have been places of teaching and learning and research. We acknowledge the important role that Aboriginal and Torres Strait Islander people play within the QUT community. So get straight into it. So Beryl still has a little bit of time at the end to wrap up. I'd like to talk to you about why acoustics is wonderful. In this image here, we have a very large, Australia's largest owl, and incredibly cryptic during the daytime, obviously being nocturnal species, very hard to monitor using traditional approaches where we have very low detection rates for surveying. And typically when we have rare, threatened, cryptic or species that occur in remote areas, we suffer with this problem of low detection rates in manual surveying. Acoustics can significantly reduce the required survey effort. We can leave these recorders out for months. These big ones that you can see in the bottom of this slide, we can leave recording for significant periods of time. We can leave them at lots of different sites at once. So we can monitor multiple sites at exactly the same time, which we cannot do with traditional approaches. And it's, as I said, a really useful tool for particularly remote areas. And I'll talk a bit more about the powerful owl in this case as a good example. It is non-invasive. I admit putting a big box could be somewhat invasive to an animal, but we try to do that in a way that doesn't interfere with the wildlife in the area. It is certainly less invasive than call playback or shining a spotlight. It provides a permanent record of the environment. We can go back in time and look at those recordings, and we may actually find things we didn't even know were there when we first set out to do the survey. And it is verifiable, so we can reduce bias because we can check it over and over again. It's not like when we release an animal or we have a sighting that we can't actually verify it again. So lots of great reasons for acoustics. That's why it's a game changer. I'm going to talk a little bit about some projects that we've been working on. One of them, and I'm just going to focus on the two birds in this project in the Gondwana rainforests of Australia World Heritage Area. So the critically endangered eastern bristlebird, which is only known from one population in Queensland. There are populations in New South Wales, but this is a Queensland funded project. And the Albert's lyrebird, which is a mimic, and its distribution in remote areas is unclear. I'm also going to talk a little bit about the powerful owl project, as I already showed you a little sneaky photo at the beginning. Its distribution and surprisingly its vocalization habits are quite poorly documented. It's a vulnerable species, and for a very large owl, incredibly cryptic. So it seems to be more common in suburban Brisbane than previously thought. We researchers at BirdLife have developed a species distribution model, and with their colleagues in the Sunshine Coast University developed a species distribution model. But as most of the records come from volunteers in the Brisbane region area, we think that probably we're missing something by not surveying the more remote areas around the greater Brisbane region. So acoustics can offer that great opportunity. Okay, so what's the problem? The problem here is that, like many of the approaches that we've been talking about today in this webinar, we end up with lots of big data. And a study recently in 2019 by Sugay et al. Has shown that pretty much we're still heavily reliant for acoustics on manual analysis. 58% of studies rely on manual analysis still. Fully automated analyses require skilled computer scientists. They're becoming more accessible for ecologists and people who don't have the great convolutional neural network brains and development skills, but it still lags behind. The amount of data we collect is just not possible to process in any other way than some sort of automated analysis. The other problem is that we have lots of low-cost tech. Audio moths, for example, you might have seen those little devices that are, quite small. Other brands also exist, and many groups are, great seizing this opportunity to collect acoustic data. But the problem is we end up with a lot of non-standardized monitoring procedures, non-standardized data. And data storage isn't considered, and acoustic and how they're going to analyze the data becomes problematic, and data curation in general. So I think acoustics is a game changer. I think I'm going to say that a few times, but we still have some work to do. So I'm going to focus now on the automated approaches that the bat world has been working with quite some time, but we're sort of lagging behind in other areas. So I'd like to talk a little bit about automated recognition. And in this project, one of the species we've worked on is the Albert's lyrebird. Just for those of you who may not be aware, we use a deep learning approach. This is the basic principle here is we convert audio to image. So you can see a spectrogram here with a call. We annotate it, we manually annotate it. We draw a box around it, that means, and we create this image that we then teach the computer to learn to look for that image. So we feed that image patch into a CNN that we've built, and then we scan through the recordings for that image. And thereby we reduce the time it takes to go through a recording, because the computer can go through and detect all of those images for us. But the sort of the catch here is you still have to have a really good training data set. And Emma mentioned this, it's the same issue you have for your camera traps. You have to have the good training data to start with, so someone has to sit down and still collect that good training data. I'm not going to spend too much time on this slide, so we can hopefully finish on time. But the point here that I will make is that training is never finished. There is never a perfect model. We will always be moving into different environments and getting, more data, and then we'd have to retrain our models by feeding back in our false positives that we might get. So it is an iterative process. So some of the hurdles. Animals do not call the same way every time, and they sound different in different locations. Geographical variation is a particular problem. So I recognise that we might build for eastern bristlebird in Queensland may not work so well down in central New South Wales. and same for the powerful owl here in Brisbane, and might not be so easily translatable to powerful owls in New South Wales. How do we detect mimics? That's always going to be a challenge. Background noise can also vary from one environment to another. Highly variable calls are really problematic. So species that have highly variable calls are going to be a challenge. And we really do need to do those basic behavioural calling behaviour studies first before we even embark on building a recogniser. There are some real easy some real problematic species for our recognisers. For the eastern bristlebird, the mistletoe bird is one that often comes up as a false positive. Grey shrike thrush for our Albert's lyrebird. Powerful owl, we've got a very low frequency call for the adult, so we've got issues with cars, dogs, and common bronzewings. So it's not foolproof, and there are things that we need to do. And it's not really just as simple as feeding calls into the algorithm. We have to tweak parameters, and we have to be careful to have a wide range of training data from a lot of different individuals and a lot of different sites. We want our recogniser to work across all environments, and so we can't have it just, we feed in only data to train our recogniser from one particular site. It might have particular background noises that, are affecting or are linked to the call of interest, and we start to see issues there. So we have to make sure that they're well tested and generalised across a bunch of different sites. We got to focus on the right call. It has to be something that's repeated, and particularly if you've got these species with highly variable calls, and we have to feed lots of negative examples or confusing species into our models. And as I said, they're never finished. You can always make it better. Just some quick results for the Albert's lyrebird. Here we have quite a high precision. So with our recogniser, which means we have a low number of false positives. It's not getting confused with a lot of different things, and not very often. We have high, reasonably good recall, and that means it's finding most of the Albert's lyrebirds in those recordings. The beauty of it is we can process 1 hour of recordings in 30 seconds. And so we've made a trade-off here. We've decided this is a good precision and recall. So we're accepting some false positives because we don't want to miss the lyrebird calls in there. So we've come up, it is always a trade-off. You have to work out what suits your research question. And Albert's lyrebirds have a lot of individual call variability, so it's going to be pretty difficult to get both high precision and recall. I will conclude shortly with this slide, and then I'll just want to add an addendum to this. Acoustic recognisers are an extremely effective monitoring tool for cryptic species, in my opinion and experience so far. We have a huge reduction in survey time. As I said, we can, have a few hours to run unattended on a deployment, and we have 20 minutes to manually verify something like 170 hours of data. So we've just, it's just going to revolutionise how we can deal with all of our big data. And one of the projects, the Powerful Owl project, for example, has a really big citizen science focus. BirdLife have their Powerful Owl Southern Queensland project, and we're pretty keen for the recogniser to be able to be given to BirdLife to be able to use to essentially facilitate their citizen science program. Challenges will always be noise. Noise is the elephant in the room for us acousticians. We don't like to talk about noise because it's very hard to deal with. Regional variation in calls, recorder variation. You might record on an audio moth, for example, but you might build your recogniser on a device or training materials from a different sort of recorder. So we have to have a look at that. Abundance is the thing we also don't like to talk about. We'd all like to be able to get abundance, but abundance estimates are really challenging to get from acoustic data. The big issues moving forward are certainly computing power, storage, data standards, data sharing, and just like to finish with this slide to say we acknowledge it is a big data science. We have project at the moment funded by the ARDC and NCRIS. It's called the Open Ecoacoustics Program, and we're basically creating a platform for storing, analysing and sharing sound data, for interoperating with other platforms, and we'd like to really promote an open source, open science agenda for this work. I'd like to just say thank you to the CSA funding grants, the Queensland Government funding grants, BirdLife and QUT, and the team, Carolyn and Lance, and Professor Paul Rowe, who's leading the Open Ecoacoustics project, and all the wonderful people who let us use their photos in this presentation. Thanks very much, Beryl. I'll hand over to you.
Thank you very much, Susan, and maybe the other panelists can turn their cameras on and their microphones so that we can do some questions and answers. And Susan, you've been talking about some questions that have come into the Q&A. For anyone that we don't answer immediately, we will follow up on the Q&As in case we don't get time here. That was really mind-blowing to hear of all these transformative technologies that are here. And I see some commonalities. Baseline data is necessary. I'm not quite sure about, Miles, do we need baseline data in your area? Or
yeah, you certainly do. So
yeah.
you do need to actually understand radar cross sections, etc. So what various animals look like in the radar. So there does need to be some baseline data done initially.
Yeah, and it's the same with TERN with its efforts to do terrestrial ecosystem monitoring. If we're going to detect change in the environment and particularly change after major disturbance, we need constant baseline data. So I presume that getting some things that I picked up, Susan, in yours, you know, it's a game changer, the acoustic monitoring, but it's got to be done in a standardized and standard protocols across everything from how you deploy, how you monitor, etc. And Emma, that would be the case with cameras as well, wouldn't it? Camera trapping. Maybe not so much for you, Alison, and the standards. I mean, but you need a common vocabulary in your area, don't you? So everyone's talking about the same thing, and you do need baseline because you've got to get everything possible identified so you can work.
That's right. Yeah.
With what you're doing. So how are we going to be able to keep this going so that we can make these technologies standard practice in conservation? Because each of you seem to be working off grants, and you know, Emma, you've had the uplift of a major disaster in Australia to be able to really demonstrate the impact of artificial intelligence and machine learning. I'm really keen as part of a national research infrastructure of how we're going to mainstream these and bring them into common practice. So panel, any thoughts?
Yeah, I can add something, Beryl. I think it's critical, and I think there needs to be national leadership on this. Essentially, we need to be developing these standards. We need to be developing these protocols. We need to have a repository. I think Alison can comment, but a GenBank is like the go-to for genomic data or genetic data. We need to have something similar in terms of sharing of recordings that are annotated so that, you know, my recordings up here can be shared with people down south, my recognizers can be shared and they can be tweaked and used. And, you know, that's how we're going to drive forward conservation in Australia, because we're all working in our little individual sort of silos, and I think that's a function of how we're funded and how things work. But I think there really needs to be a push forward on this at a national level.
From a radar perspective, briefly, at least using weather radar, that's infrastructure that's already there. That's government infrastructure, so that's something that people can tap into and use. And it's being used quite successfully already in Australia for, say, monitoring fruit bats or birds. The other units, or the more portable units, they do require an infrastructure cost to be laid out. But the good thing there is that if many people were to, say, step in and buy a series of units, you could start to network them and everyone could get benefit from that data. So again, it's a matter of data sharing and standardization, but it's certainly something that would be achievable if the funding can be generated and people would actually work together. I think a bit more to link in.
Yeah, I wanted to just echo all of that. I mean, not just what you said, what we've said here today, but in the 2021 State of the Environment report, one of the big things to come out of that was a need for more monitoring and to do things better than what we're currently doing. So nationalising that, working with groups like TERN, for instance, like what we're trying to do through Wild Obs, is really important. Developing standardised methods and you know, basically trying to get this data out there through a lot of collaboration. As researchers, we like to kind of grab onto our data and not share it sometimes, because we like to sort of, you know, publish, and that's a competitive thing for a lot of academics. But I think we need to sort of move past that, like a bit of a call for action. Let's start working together, not just think of our own research and our careers, but think of the conservation outcomes that need to happen to basically address these big threats that we're currently facing, like, you know, climate change, all of that sort of stuff.
And on that point, I think I'm going to say thank you to all of our speakers, and you've all been a wonderful audience, because I can see from the chat and the questions and answers that you've become quite involved in these transformative talks. So we look forward to seeing you again at our next webinar, which is all about natural capital accounting, and I think you'll find that really fascinating, on the 2nd of November. So thank you, speakers. Thank you, audience. We'll see you again sometime soon.